Pharmaron's 40,000 square foot clinical pharmacology center is in downtown Baltimore and is part of the University of Maryland Biopark. The university's medical center is across the street and is the closest emergency room. Whether coming for screening or to check into a study, all participants are met in our outpatient screening area. For those in screening, the informed consent process is initiated through telehealth consenting before arrival. Study-specific screening activities are conducted once the informed consent form is signed. All potential participants are examined in one of 4 exam rooms. Exams are performed by the study principal investigator or a sub-investigator. When participants begin a study, their street clothes, valuables, and non-essential belongings are documented and secured in locked storage units. The inpatient units have shower facilities and bathrooms that are easily accessible. Depending on protocol requirements, access to these facilities may be controlled by clinical staff. To ensure safety for our participants, team, and trials, security cameras are located throughout the facility. In addition, all doors on both the clinical research unit and offices are keycard access controlled. There are 90 beds, and dorm sizes range from 3 to 20 beds. Study participants are closely monitored throughout dosing and clinical activities. Sponsor representatives can observe dosing here. Our trained clinical staff ensures participant safety. All inpatient study activities, such as blood draws and ECGs, are conducted at the bedside. Our staff explains all assessments to study participants. Satellite set and synchronized digital clocks are throughout the site to document all timed activities. Participants can relax in one of 2 large recreation rooms, which include a pool table, internet, gaming systems, and TVs. Participants receive 3 catered meals plus 1 evening snack. A nutritionist provides consultation on meal options or special diets. Our data management team sets up in the nurses' station and enters data as close to real time as possible. Each unit has emergency equipment, also referred to as a crash cart, which is checked regularly by nursing staff. All nurses and investigators have advanced cardiovascular life support certification, and all clinical staff are BLS certified. The pharmacy has total controlled access and provides on-site sterile and non-sterile extemporaneous drug compounding, including highly potent IP. As part of our human carbon-14 metabolism studies, our pharmacy team prepares clinical dose and conducts formulation assessments to ensure unit dose accuracy and to support our sponsors' regulatory updates. All investigational products are stored under appropriate environmental conditions in a secure location in the pharmacy. Each study has a designated locked cabinet with a study binder and supplies. Temperature in the pharmacy area, refrigerators, and freezers are monitored by the Reese temperature monitoring system. All equipment, counters, work surfaces, and floors in the ISO Class 7 and Class 8 area are cleaned daily during business days and before and after compounding sterile preparations. In the pharmacy anteroom, all refrigerators and freezers are locked, and the temperatures are monitored by Reese. These reports are printed and checked daily during business days. All pharmacy equipment is inspected prior to use and per manufacturer's recommendations. Written records of inspection and calibration are maintained on site for 2 years, then moved to a storage facility. Pharmacy staff are required to wear PPE and wash hands according to USP 797 and Pharmaron standard operating procedures before compounding sterile preparations and routine cleaning. Clinical labs covering tests for hematology, blood chemistry, coagulation, urinalysis, and study-specific biomarkers are run through state-of-the-art instrumentation. Our staff adheres to best practices and biosafety guidance when working with biological specimens in the post-COVID-19 lab setting. This instrument uses fluorescent flow cytometry and cell counting methods for rapid hematology panel analyses. The Abbott Alinity analyzes chemistry and immunochemistry and is among the highest-rated clinical chemistry analyzers. We have AlinIQ AMS, which fully automates specimen labeling and tracking and also interfaces with the electronic capture of clinical data. Additionally, the lab facility features both refrigerated and unrefrigerated centrifugation. We collect and process PK samples, including carbon-14 for our sponsors, which are then analyzed by our bioanalytical laboratory. samples depart the clinic and arrive within 1 hour at our bioanalytical laboratory. Clinical project managers work closely with our team of bioanalytical scientists to facilitate efficient, quality data delivery for all studies conducted at our Phase 1 unit. Samples are logged and tracked from receipt to disposal using a 21 CFR 11 compliant electronic laboratory information management system. Biological samples for PK, biomarker, radio-labeled ADME, and metabolite profiling studies are analyzed by expert teams in our GCP-compliant laboratories. Our medical writing team is an integrated part of our clinical service, which includes preparation of the protocol before the study and the CSR after the study. Our integrated early clinical development services enable our customers to rapidly deliver insightful data.